Okay, so welcome to this next video in which we're talking about the adaptive immune response. Okay, so at the moment what we're doing is going over the innate immune response, also known as the acute inflammatory response, just to have a complete picture basically as to where the adaptive immune response fits in, because often the innate immune system is taught completely separately from the adaptive immune system, and people struggle to put the two together and get a complete picture of the immune response. So we're just going to briefly go over the acute inflammatory response. And we're nearly there now. The release of these three cytokines was really what triggers the acute inflammatory response. Okay, so we discussed that when sentinel cells, such as dendritic cells or resident macrophages or mast cells, sense general molecular patterns associated with pathogens, known as pathogen-associated molecular patterns, they become activated and they're going to release pro-inflammatory mediators. So the dendritic cells and the resident macrophages are going to release interleukin-1 and tumor necrosis factor alpha, and the mast cells are going to degranulate and release histamine. Now, this is going to have an effect on the blood vessels within this area, basically. Okay, so you'll have three types of blood vessel that are within this area, basically. You'll have arterioles, then you'll have capillaries, and venules, finally. So let me just draw this. So here is a small little arteriole, and you really should understand that arterioles are tiny little blood vessels. Do not think of them as being anything like arteries. Arteries are those massive great things that you have to learn about in anatomy. Arterioles are tiny little blood vessels, but they do have smooth muscle cell lining them still, and that's why they're going to be important here. But they are tiny, so you'll have these in your little tissue. Okay, then it'll split into even smaller blood vessels, and I'm only going to draw three of these. Okay, so it splits into these tiny little blood vessels known as capillaries, and capillaries are the business end of the microvasculature, and all of the blood vessels I'm going to talk about are considered microvasculature. Okay, the macrovasculature is the stuff you learn about in anatomy. Okay, so they're the business end of the microvasculature because this is where the exchange of nutrients, so the exchange of oxygen and glucose out of the blood and the waste products coming in, such as carbon dioxide, it's going to occur at the capillaries. Okay, so now we've got our three separate types of blood vessel. The arterioles here, and the important thing to understand about the arterioles is that they have a smooth muscle layer. Okay, then the capillaries, which literally are tiny, they just have an endothelial cell layer, okay, with a basement membrane for the endothelial cells to sit on. And then finally the venules, and the venules I would encourage you to think of just as bigger capillaries. They again just have an endothelial cell layer with a basement membrane, so the wall of a venule literally consists of an endothelial cell sitting here, okay, with a basement membrane which mainly consists of collagen that it sits on, because it has to have something to support it. Why does it stay in its position? Well, because it's attached to the basement membrane. Okay, so in turquoise here, this is the basement membrane. And let me just make sure this is still in view. Excellent. Basement membrane. Right, so both capillaries and venules only consist of a basement membrane with endothelial cells around them. Arterioles are a little bit more. They'll have a smooth muscle cell layer around the basement membrane, so they're slightly bigger, but, well, as far as the wall is considered, they're bigger. Okay, so venules are just like a bigger capillary. Capillaries are tiny. They only have a lumen that will allow just one red blood cell to barely squeeze through, whereas venules are a little bit bigger. You know, they'll allow multiple red blood cells at least to fit within them. Okay, right. So, what's going to happen then? Well, the tumor necrosis factor alpha, the interleukin-1 and the histamine are going to go and act on these three types of blood vessel that you have within your tissue, okay? And they are going to lead to the innate immune response, or the acute inflammatory response. And this response consists of you bringing things out of the blood into the affected tissue, basically. Everything comes from the blood. You are going to bring out troops from the blood and bring them into the interstitial fluid, which is just a fancy name for tissue fluid. Okay, so the fluid that bathes the cells. Interstitial fluid. Okay, and they are going to fight this pathogen which has decided so foolishly to come and live in our tissue, basically. Okay, so what's going to happen? Well, histamine is going to act on the endothelial cells that are in the arterioles, the capillaries, and the venules, and so is interleukin-1 and tumor necrosis factor alpha. However, they trigger a different response in the endothelial cells. Histamine is going to trigger what's known as type 1 activation of the endothelial cells. Okay? So histamine is going to trigger type 1 activation of endothelial cells. Now, this occurs within minutes, basically. Okay? And what will happen is that the endothelial cells will start producing molecules which will cause vasodilatation of they'll cause relaxation of smooth muscle cells. Okay, so let me repeat that. Histamine will cause type 1 activation of endothelial cells, and in type 1 activation of endothelial cells, the endothelial cells start producing vasorelaxant molecules which are going to cause relaxation of smooth muscle cells. Okay, now, is this going to be important in the capillaries and the venules? Well, no, because they don't have any smooth muscle surrounding them. However, in the arterioles, this is incredibly important, because what it's going to lead to is relaxation of the smooth muscle cells surrounding the endothelial cell layer of the arteriole, and this will then lead to the relaxation of that, the relaxation of the smooth muscle cells will lead to the widening of the blood vessel, because basically, if I just explain this, so if we draw a little arteriole here, okay, so let's say this is the basement membrane in turquoise here, and you'll have the endothelial cells sitting on the basement membrane. Let me show these. So here's an endothelial cell sitting on the basement membrane, and obviously you'll have a complete ring of them, like so. Okay, so a few more. Okay, there we have the endothelial cells. Then surrounding the basement membrane, what you then have is smooth muscle cells, vascular smooth muscle cells. And these vascular smooth muscle cells are arranged in rings, basically. So you have rings of smooth muscle cells around the arteriole. Now, what's going to happen if each one of these smooth muscle cells relaxes? Well, basically, it's going to get longer. Every single smooth muscle cell will get longer. And if every single one of them gets longer, then that means that the circumference of this entire ring will get bigger. Now, if the circumference of a ring gets bigger, what's going to happen? It's going to become a bigger ring. So if you imagine, how can I show this? There's probably better. If you imagine the circumference becoming bigger, it becomes a bigger ring. The diameter of the ring also gets bigger. Okay, so the rings of smooth muscle cells are going to dilatate, is the fancy name for the diameter getting bigger. And when that happens, what will also happen is the diameter of the endothelial cell ring and the basement membrane will also dilatate, and basically you'll widen the lumen through which blood is flowing. So this is going to cause vasodilatation when you relax the vascular smooth muscle cells around the arterioles. Okay? And vasodilatation is the correct word for vasodilation. Vasodilation is kind of a shorter word for vasodilatation. Okay, right. So you get vasodilatation of the arterioles, okay, which leads to a greater blood flow to the affected area. So when you widen these arterioles, you're going to get more blood flowing into the affected area. So blood flow goes up. That causes the affected area to become red and also to become hot, okay? Because blood is red and blood is hot. Okay, right. So what's the purpose of vasodilatation? Well, it's to bring in more blood so that you can bring in more troops that are going to fight the pathogen. Okay, so let's talk about what else type 1 activation does. In addition, what it leads to is endothelial contraction. Okay, so I'll put contraction there. And this should not be confused for contraction of the smooth muscle cells. Obviously, you've just relaxed the smooth muscle cells. No, this is endothelial cell contraction. So what's going to happen is the endothelial cells, okay, so if I draw another picture like this over here, okay, so basically, if this is an endothelial cell here, okay, then it will have a neighbour sitting next to it. So let me show its neighbour. So here's its nucleus, and here's its neighbour. And basically, the endothelial cells, at this junction between two neighbouring endothelial cells, they have protein complexes holding the two of them together. Okay, so let me bring this up a bit. Okay, so I'll draw the basement membrane on the bottom here, which both of these endothelial cells are sitting on. So basically, at the adhesion between the two endothelial cells, you have two protein complexes that are holding these two endothelial cells together. The one that's closer to the lumen, is known as a tight junction. Whoops, what am I doing? Tight junction. Okay, now I can't fit in junction. Tight junction. There we go. And then the one that's closer to the basement membrane down here, that's known as an adherens junction. Okay, right. Now, these junctions, these complexes, form a physical occlusion to anything moving through the gap between the two endothelial cells. Well, firstly, they keep the two membranes of the endothelial cells very close together, but also the proteins that are actually involved in these two protein complexes, they form a physical occlusion to molecules which may well try and move through this gap between the two endothelial cells. Okay, so in endothelial cell contraction, what happens is you pull, each of the endothelial cells pulls its proteins that are involved in each of these junctions away, basically. So to explain this in a bit more detail, I'll call this endothelial cell 1 and this endothelial cell 2, and I'll go over the page. Right. Okay, so if this is the membrane of endothelial cell 1 here, and this is the membrane of endothelial cell 2 here. Okay, so let's call this endothelial cell 2, and this endothelial cell 1. Then basically, endothelial cell 1 will supply a bunch of proteins that are involved in the formation of this tight junction here, and so will endothelial cell 2. So these are the proteins that are involved in the tight junction. And half of the proteins are supplied by endothelial cell 1, and the other half are supplied by endothelial cell 2. Okay, and it's the same case, well, the same thing is true for the adherens junction. Half of the proteins are supplied by endothelial cell 1, and half by endothelial cell 2. So basically, what happens in endothelial cell contraction is that the cytoplasmic domains of these proteins that are involved in the tight junction are attached to actin filaments. Okay, so they're attached to actin filaments, and the same is true for endothelial cell 2. Okay, so the cytoplasmic domains are attached to actin filaments. So these are actin filaments. Okay, so I think I might colour the actin filaments in green. Okay, so here are the actin filaments now coloured in green, which makes them look a little stronger, a little less whoops. Okay, that one's going to be a massive great actin filament now. Right, okay, And it's the same case for the adherens junction. They have actin filaments attached to their cytoplasmic domains as well. So what's going to happen in endothelial cell contraction is that you're going to contract these actin filaments, okay? And basically what's going to happen is each of the actin filaments, when they contract, for instance, if we take this actin filament, it's going to pull these proteins in this way. This actin filament's going to pull these proteins in this way. This actin filament's going to pull these proteins in this way, and this actin filament here is going to pull these proteins in that way. And you can see that this is going to pull the tight junctions and the adherens junctions apart. And that's going to open up a gap in between the two endothelial cells. Yes, it's a reasonably feeble gap, but you have got a gap nonetheless between the two endothelial cells. Now, this type of endothelial contraction is going to occur in the capillaries and also the postcapillary venules, okay? So basically, what it's going to lead to is little gaps between the endothelial cells, and this is going to mean that fluid from within the blood can move through those gaps between endothelial cells and go into the interstitial space. In addition, proteins that are in the blood can move through the gaps between the endothelial cells and into the interstitial space, okay? So you bring in fluid and proteins from the blood, okay? And this is what's known as an inflammatory exudate. I also want to stress that these gaps that you create are not strong enough for, are not big enough for cells to move through. They're big enough for fluid and proteins to move through, but they're not big enough for cells to move through, at least not without help. Okay, so you will form an inflammatory exudate, which is basically made of this extra fluid that you are allowing out of the blood that would never usually get out of the blood and into the interstitial fluid. Right, so it's going to lead to swelling of the affected area, basically. Now, what's the purpose of this inflammatory exudate? Well, the purpose of the inflammatory exudate is to bring in proteins that are within the blood plasma into the affected area, okay? So, two major protein types that you're going to bring in are you're going to bring in complement proteins, which can attack pathogens, basically, through three complement cascades, known as the classical complement cascade, the alternative complement pathway, and also the mannose-binding lectin complement pathway. Now, the classical complement pathway actually requires the adaptive immune system to work, and we'll come back to that later. But the other two, the mannose-binding lectin pathway and the alternative pathway, both will occur instantly, basically, as soon as they see pathogens. Okay, and they will lead to the destruction of those pathogens. Okay, and also to bring in the coagulation factors. Okay, and the coagulation factors are proteins that are produced within the liver, and basically, when you allow coagulation factors out of the bloodstream and into interstitial fluid, then they set off the intrinsic and extrinsic coagulation cascades, which leads to the inactive protein fibrinogen being converted into fibrin, which is then assembled into fibrin strands. So basically you produce a huge number of fibrin strands, and these fibrin strands form a meshwork, basically. They form a fibrin meshwork, which you can imagine as a very dense spider's web of fibrin strands. And this is going to intertwine amongst these pathogen cells, or pathogen, if we've got viruses, they're not really cells. They're going to intertwine among the pathogen structures, okay, and contain them, basically, and try and stop them from infecting, from spreading. Okay, right. So this is going to actually attack the pathogen. The coagulation factors are going to produce a fibrin meshwork that is hopefully going to prevent the pathogen from spreading to other portions of the body. Okay, so that's the purpose of formation of this inflammatory exudate. Okay, so we'll continue this discussion in the next video.